far, we've been looking at the behavior of bacteria and things like that. So in the previous lecture, we talked about how their dynamic steam flow causes infections and how we can use physics to prevent that by making a tube with corrugations in them that would deflect them, but also the flow generated at these edges also help them go downstream. So now we want to sort of build up a model of how the flow these bacteria is going to generate when they are in water, okay? So let's think about their motion slightly for a bit. Okay, so because these guys are small, so they're always undergoing Brownian motion. So here's a experimental video of what let's call it Climatomolus that's moving in this direction. So if you think about these micro scale systems, their typical size is about on the order of microns, okay, 1 to 100 microns typically, and their speed of motion, speed of self-propulsion is often on the order of the 1 body length per second. And that is true across length scales, okay? So if you're thinking about humans, running the fastest you can run is about, at most 2 meters per second. That's all you can do. So what's important for physics of these swimmers is we want to consider what's called a Reynolds number, which compares the inertial forces to viscous forces. So here, U is the typical speed of the particle or a swimmer. L is their body length. And this nu is the kinematic viscosity. So that's the dynamic viscosity, but scaled by the density of the fluid. So if you put in these numbers and put these swimmers in water, this is a Reynolds number you're going to get. Very small, much smaller than 1. What that means for physics or for Newton's law of motion is, first, we can neglect the left-hand side, which is the inertial term. But also we're talking about self-propulsion. So these swimmers, they're generating their own thrust. So we're not applying an external force to this system. So there's also no external force. And the only, because we're talking about self-propulsion instead of applying a force, then dragging this bacteria along. There can be, if you apply a force, then there is. Yeah. For now, yeah. Yeah. For now, if you talk about self-propulsion, there's no force. And the only force left is a net force from the flow. That's called the hydrodynamic force. What that means is this total hydrodynamic force is 0. And then the point is, how do you get your drag and your propulsive force? They all come from this hydrodynamic force. Okay, so you can break that up into 2 pieces. One is the drag force. One is the swim or the self-propelled force that you're getting, but that they all add up to 0 at all times at these scales. So that's a very interesting observation. And then if we think about flow at 0 Reynolds number, so have you heard of the Navier-Stokes equations? Show of hands. Okay, most of you. So the Navier-Stokes equation just describes, that's a Newton's equation of motion, okay, but written for a continuum field. That's why it looks a bit more complex. And also, this is written for a Newtonian fluid, where you have a dynamic viscosity mu, and we also have a continuity. That's just due to the fact of conservation of mass. So when the Reynolds number is small, so in this equation, the Reynolds number is essentially comparing the inertial terms that's on the left-hand side to the viscous term on the right. So that's a scaling for the Reynolds number. For these bacteria and the flow they generate, this Reynolds number is very small, much smaller than 1, which means at leading order, the most important effects are on the right-hand side. So that's a balance between the viscous force and the pressure gradient. So these equations are called Stokes equations. You have momentum balance and continuity. Continuity is unchanged. So there are some consequences of Stokes equations. If you look at these equations, what do we see? They're linear equations because we have removed the nonlinear term entirely. Now, if you think about a particle or an object, say that's translating at a velocity u in a viscous fluid like this, say in the first case, this particle is translating to the right-hand side, okay? And you can solve these equations to figure out the flow field in entire space. What happens if you now have the particle translate to the left with the velocity minus u? What happens to the flow field? Because the equations are completely linear, the flow field simply reverses direction. That is it. Okay, that's a consequence of linearity, which means when we think about an object, a rigid object that's moving in a fluid, either translating with the velocity u or rotating with some angular velocity omega, there is a linear relationship between the force or the external force that you applied to drive that motion and the velocity you're getting. So that constant of proportionality, which we call the resistance tensor previously, for a single particle in general, you can write it this way. You have both the linear and angular velocity. And for the forces you applied, you can have external force and an external torque. So if you write down that tensorial quantity, you have 4 terms here. And the first term here essentially couples the force to the velocity. The second is force to the angular velocity. And similarly, this one is written wrong. That's Lu. Okay, that's flipped anyway. So what's interesting about the resistance tensor is it's actually symmetric, and that's a consequence of these equations, which we're not going to talk about. You can prove that.
So in this way, this force as well as the start both are connected closely.
Which one?
Force and the torque.
Yeah.
The force and the angular.
In angular velocity, in general, it can be, and then we'll talk about in just a moment of what. Yes,
as really expressed here.
Yeah.
They are connected closely.
Yes, essentially. So in general, if you apply force on an object, it can rotate. When does that happen is something we'll think about in a moment. But in general, that can be allowed. Okay, and that's allowed is for a chiral object. We are driving a screwdriver. You are spinning the screwdriver, but the screwdriver is translating. So that's a translation rotation coupling, and that's reflected here in this off-diagonal terms. Okay, you apply force, it's going to rotate. You apply torque.
This equation is why you this seems to be in the form of a radiation.
This one? And this is actually a diffusion equation. So we'll talk about it in just a moment of how that behaves. So essentially, we talk about Stokes equations. We're just solving Laplace equations and Laplace. Yeah. And the nice thing about that is there is very nice structure to it, and then that also allows you to compute these things easily. Like use as a fundamental for computers. And so for chiral objects, you can have translation very powerfully. For sphere, it's isotropic. If you apply a force on a sphere, it's not gonna rotate. Okay, so which means these terms are gonna be 0, and these are isotropic. Okay, that's the Stokes drag, and that's the Stokes drag in the angular degree of freedom. So that's the torque you get by rotating with an angular velocity omega. Okay, so these tensors are functions of the geometry of your object. So if you know the geometry, in principle, you can calculate these terms. Of course, in practice, you compute them numerically sometimes can be tricky. Yep.
Then this is what?
This is a single particle in a Stokes law. Yeah. And other comments?
Yes.
Okay, that's great. So if we come back to biological swimmers, here we're looking at an example here. So what happens in this case is this microorganism has many tiny little hair-like structures that covers the entire surface. If we look at the fluid velocity in layer that's right outside these hairs, what we can do is the velocity here must be equal to the velocity of the object. That's the rigid body translational velocity of the swimmer at this instant in time, and an angular velocity. So in general, it could be rotating, plus the addition term, because these little hairs here are generating local flow around the cell. So you need to add that, let's call it sleep velocity there, that's generated by these little disturbances. So around this red surface, the fluid velocity can be written in this fashion. And this last velocity is contributing to this propulsive force. And these, when the particle is just translating and rotating, then that gives rise to a resistance or friction. So if you add these up, they have to be 0. But then, you can invert this relation to give the velocity in relation to the propulsive force you're generating. So that's how, in general, a particle or a swimmer is able to self-propel in a Stokes flow. Okay, so essentially for a sphere, that reduces to friction coefficient times the velocity you're getting. Okay, that's great. So there are other kind of swimmers. So usually we classify them into two types. One is called a pusher, one is a puller. So a pusher is things like E. coli. So it has a cell body in the front and a tail. So the tail is pushing the cell body forward. And that's why it's called a pusher. Whereas for this organism here, Chlamydomonas, it has sort of two flagellum in the front. So it's dragging the cell body forward. It's pulling this forward, so that's called a polar swimmer. And if we think about these swimmers and where do they generate their propulsive force and where their drag is coming from mainly. So if you think about a pusher, the cell body here is largely contributing to the drag. Okay, all the propulsive forces are coming from the tail. So if you think about the force that's exerted to the fluid, that's the red arrows. So for the tail, it's going in this direction because the propulsive force you're getting from the fluid is to the left. Okay, so that's the force onto the fluid. And similarly, the force exerted on the fluid by the cell body is to the left. So if you zoom out, if you look at this structure far away, that looks like a force dipole, which is just two forces aligned, but very close together if you zoom out. So you would generate this kind of flow field. So it's pushing fluid out from the front, right, and the back. Then by conservation of mass, the fluid has to come back from the side. So that's the blue arrows there. And they're, in this case, they're swimming in this direction. So that's for pushers. Pullers, like Chlamydomonas, is behaving this way, okay? So the direction of the force is reversed, but nonetheless, they're again balanced for self-propulsion. In this case, they're drawing fluid in from the front and back. And then again, by conservation, the fluid has to go out on the side. So there are two different kinds of swimmers in general at this scale. And the point is, to what extent can we represent the flow field these guys are generated using these force dipole approximations, which we know intuitively are only good when we're looking at it far away, right? Far field. So here's some experimental measurement of the flow field. So here's a E. coli or a pusher kind of swimmer. So on the left-hand side, that's the flow field is generating, okay, measured from experiments. And here on the right, the top panel, that's the flow field you get by looking at the flow is generated by a force dipole like this, which you can compute analytically and plot it here. And here is the difference between the flow field due to the force dipole and the actual flow field. It's half of it, okay? Because it's the top and the bottom are symmetric. So the difference, if you look at this graph here, of course, the highest error comes when you're really close to the swimmer. Then you can see actually they're not a force dipole. They're well separated. But if you're far out, the difference is minimum. So it's a good approximation if you're far away from these swimmers. Okay, any questions? Okay, yeah.
But the one here is the biggest thing.
Words, yeah.
You could take the swimming of long pieces actually.
In this case, we're not talking about the rotation yet.
Senses.
The
Axis.
in real life they can rotate, yes. Because these two flagellum can be misaligned, then the force it generates, again, it's slanted, so they will rotate, yes. We will incorporate that later on into our models we're trying to build, but for now we're just looking at simple force dipoles and how the velocity they generate the flow they generate looks like. Yep. Other comments? Okay. So what I want to think about now is rheology. So when we have bacteria in water, how does that modify the rheology of the whole system? So rheology is, or we're using a rheometer to measure rheologic behavior, is to quantify how general materials deform and flow under applied stresses. So usually what we want to do in this context is to determine the stress and the strain rate, or if you're doing a shear rheometry, the shear rate. So here's a typical tabletop rheometer this is a cone and plate rheometer. Okay, you have a cone here and a plate. You put the fluid sample in the middle, and you by rotate this device, you're going to shear the sample in the angular direction. Okay, so you can apply a definite shear rate, and you can measure the force you're getting from that. And that's a typical experimental measurement you can do. And using rheology, you can quantify materials generally into different categories, especially for fluid medium. So here's a stress shear rate graph. And the one we're most familiar with is the Newtonian fluid, which means the slope of this curve is a constant. Doesn't depend on the shear rate. Okay, that's the Newtonian fluid. And you have other kinds of fluids, then those are called non-Newtonian. You can have shear thinning fluid, which means if you the shear rate is higher, the slope actually goes down, right? It becomes easier to flow.
I think the slope
The slope
slope is
is the derivative of y with respect to x.
the city because when you were the
For Newtonian, yeah. For non-Newtonian, the viscosity or the slope is a function of the shear rate. So yeah,
so he's saying that you know this
that's right. Which means you get a flat or a straight line. Okay. You have other materials. One is interesting is shear thinning, which as you shear faster, the slope goes down, right? If you look at this, the slope is smaller. And the opposite of that is a shear thickening fluid. As you shear faster, the viscosity goes up.
Yeah.
So that's interesting. You have other kinds of materials. We're not going to go into details. You have Bingham plastic materials. What that means is there's a definite stress before you can move. So there's a yield stress. If the stress you have is less than that, it's not going to flow. There's a critical stress that you need for it to flow. And once it flows, you can, again, you have other models, but the ideal Bingham model would be, looks like a Newtonian once you cross the threshold. Okay, and you have many other kinds of materials. And we've seen this kind of, so these kind of materials are quite common in daily life and in applications. So here's a video from YouTube for a shear thickening fluid. So let's look at this video and see what happens here.
Do you know what happens when the is a special type of substance that grows stronger when faced with force and weakens when the pressure is reduced, to harness the variable viscosity properties of non-Newtonian fluids to design lightweight armor and absorbing safety systems. This lab first suspends the non-Newtonian fluid over the trap using a rope, and the moment the trap is triggered through the slow motion lens, we see the fluid instantly transforming into a solid.
So this is not just any non-Newtonian fluid, that's a shear thickening fluid. Okay, if you apply the force very fast, the viscosity blows up essentially. So it behaves like a solid. However, if you push it slowly, you're going to sink into the fluid. So that's a shear thickening behavior, okay? And it has applications. So in general, you have all kinds of non-Newtonian behaviors. We talk about shear thickening fluids. So the one we watched in the video, that's essentially cornstarch and water mix, which is shear thickening. You have plastic materials or shear thinning fluids, such as ketchup and so on and so forth. So you have other kinds of rheological behaviors. So these have many applications. For example, in oil drilling, typically you use it depends. So in this, not it depends. Sometimes it's more plastic, Bingham plastic. So if you don't apply any, okay, so if you just have squeeze out the toothpaste, you put it there on the table, it's not gonna flow, right? So it doesn't flow under zero. Yeah. So sometimes it's
something which we already can work.
Yeah. That's right. So you can play with that. That's the video we watched. That's essentially a cornstarch and water mix.
It's part split.
Yeah, Woolpack. It has a specific name for it. Yes. So you can jump on it. So long as you jump fast enough, it looks like a solid. Okay. So that's very nice. And so these fluids have many applications. For example, in oil rigs, you would have drilling fluid. And those are often shear thinning because you want them to flow easily. And when you don't shear them, you want them to maintain a certain structure so that the things you get are not going to easily sink down. And you can, for body armor, for example, you would use shear thickening because when something comes very fast, it behaves like a solid, okay? So these have many applications. And the point is, when we think about active suspensions or bacteria in water, what kind of rheology are we going to get? So here is an experiment done with, these are different bacteria concentrations. They're fairly dilute here. So we're measuring the viscosity of the suspension, which means bacteria in water, okay, as a whole system, compared to the viscosity of water as a function of shear rate. So what we see here is when bacteria density is very low, of course, you're pretty much close to unity. That's the viscosity of water. As you gradually increase the concentration of the bacteria, what we see is the viscosity is lower, okay, in the zero shear region. Of course, again, the reason that these curves collapse for...
the concentration of bacteria's viscosity decreases.
Yes. Yeah.
Okay.
If it's dead bacteria or passive particles, it will go up, right? But not for a bacteria, it goes down, and we'll talk about why that's the case. But essentially, if you look at this regime, which is easy to understand, so in this regime, the flow is much stronger. The flow generated by the rheometer, okay, you're turning the rheometer, which induces flow. So in this regime, as we talked about before in the previous lecture, when the flow is very strong, you can tell really whether these guys are swimming or not. So it behaves like passive, which means here the viscosity is higher than water. You have some passive objects in there, essentially. But when the shear rate is low, I really can tell whether these particles are active or not. So in that case, activity is important, and in this situation, the viscosity is brought down because of activity. And why is that? So there's a sort of a simple physical understanding of it. So in a rheometer, we can sort of approximate the flow generated as a simple shear flow like this, okay? So if we take this fluid velocity profile, we can decompose it into two components. One is rotating, because you're shearing, which will induce rotation in this direction. So that's the first component. Now, if you remove that part from this flow, what you get is an extensional part, okay? So now if you think about these bacteria, so in this case, this is done for E. coli or a pusher-like swimmer. So these pusher swimmers are, again, they have, to first order, there are 4 cycles, but they're also non-spherical, okay? So you put these swimmers in this extensional flow, they get aligned along this axis because of their non-spherical shape, okay? That's what happens regardless of activity. Non-spherical particles in the flow will tend to align with the extensional axis. But once they're aligned, these guys are active. They will generate their own disturbance flow in the fluid. For a pusher, their force dipoles are in these directions. So the flow they generate is also an extensional flow in the same direction as the flow you have applied. So what does that mean? It means that you're trying to generate this flow, but the bacteria in the water is helping you. And it's also generating this flow. So you don't need so much effort to induce that flow, which means your viscosity is going to go down. Okay?
Just give it away.
So this is the flow you're applying. Okay? But the bacteria generates the same kind of flow. So you need less effort to produce the same amount of extensional flow because someone is helping you.
So yes, because the rheometer is measuring less viscosity, right?
Yeah. Because the flow you generate, so this is contributing to the flow you're generating, right?
Rheometer also applies.
Yeah, that's what this is, right?
But now the bacteria is applying less.
Yeah, so to do the same amount of flow, I need less force because the bacteria is generating the same flow, right?
In the fluid.
In the fluid, yes.
In the rheometer is applied.
Yeah.
That force is measuring the viscosity.
That's correct.
But will not for I'm just saying I'm confused.
Yeah.
That viscosity because how the rheometer measures the viscosity. That is why this kind of thing helps the tree.
Sir, could you?
Rheometer is measuring the viscosity.
The viscosity. Yeah.
It's not doing it's not doing some trick.
So the viscosity is the real thing that exists.
It measures
That's correct, because the viscosity is defined as a force, so the ratio of the force or the stress and the strain rate, right?
The viscosity flows. It seems like the fluid has both kind of viscosity.
What do you mean both kind of viscosity?
Like this, talk to us that like force, right? This spectacle.
Yeah, the bacteria exerts a force in the fluid, right? Because it's swimming around, it disturbs the fluid. That's precisely the point, because the bacteria in the water is applying force which generates a flow. Therefore, the viscosity you measured for the system is different from water now. Yeah,
It is coming down like
it's coming down.
So,
and that's what you're measuring is that measuring the actual.
Yeah,
why is the measurement is not interfering with the
Yes, yes.
yeah,
This the point is that what Oscar is asking is that the hydrometer is rotating
yeah,
and somehow it affecting the like
that's correct. Yeah, yeah.
the fluid itself is having the bacteria, which is actually changing its viscosity.
That's correct.
How much viscosity has been changed is being measured by the
That's right. Yeah, yeah. That's all. That's it. Yeah.
same viscosity is not changing. No, viscosity.
But now it's the viscosity of the suspension of the fluid plus bacteria as a whole, not just the water, right? Yeah.
It is not like quantum diversity.
No.
There is an observer which is checking the behavior of the particle. Isn't that the case? So
Yeah.
classical system, you are talking
Mhm.
classical system where instrument is measuring what is there.
Okay. Other comments? So that's for pushers, right? And for the other type of bacteria, pullers, the flow is in the opposite direction. So you're actively resisting this motion I'm generating, which means if you were to do an experiment for pullers, you're not going to get a less viscosity. So that's why this is done for E. coli, where only for those kind of bacteria you can get a reduced viscosity in this kind of experiment.
should bacteria look to
This is pusher. This is puller. Yeah. It's this one. This one.
Yes.
Yeah. Or if you look at the cartoon, this kind of bacteria. So essentially for this bacteria solutions, the viscosity is going to be lower than a solvent. For this, it's going to be higher.
so these sperm activity percentage changing. These are pullers.
These ones are pushers.
Sure.
Yeah.
Now they in this concentration. Sorry, the different percentage of activity.
No, those are just the concentration, number of bacteria. Concentration. Yeah.
So
In water. No. Y-axis is viscosity. Concentration is different colors and shapes in the curves. Yeah.
So you see the concentration is actually from start to down.
Yeah.
1.7, 1.2, so it is increased.
Yeah.
Viscosity is coming down.
Yeah.
The viscosity is coming.
Yeah.
makes it.
That's right. Yeah.
So you got more than you have more.
More agents in there are doing this. Some of them are viscosity down for as an increase in concentration.
Well, if not, yeah, still we don't still that will still was us. Yeah.
Suppose you have this kind of bacteria and they are in a level and ocean. And now they are rotating putting them on the rotating table. So where the superficial motion the table itself is going to affect the area. Then the nature the shape is for example the fluid is in the land and so in certain concentration of bacteria so viscosity is running the whole. And so now the fluid is rotating. Now so liquid is rotating, whether with characteristic of the bacteria that it pushes will really change and because that means viscosity will change.
Yeah. So you know about this kind of form. The bacteria won't be aligned. So they're just random. But then you can't measure viscosity if you don't apply a force. Okay, sort of composite structure, right? Motors linked to microtubules. So often that term is quite small in this particular context. So we don't have to leave that here. We can neglect this term. Neglect the question. So we're specifically modeling the motor protein microtubule kind of pneumatic system, right? Here. And in that case, when you talk about particle, the particle is a composite particle. It's made of microtubules connected by motors. So that thing is not very diffusive. If you measure specifics, it's quite small. So to medium order, we don't care about this term. It's quite weak, that's all. And then we just leave it out of the equations. And then what we have here, then we have this term here, right? And we look at this term here, we can distribute the divergence. What's the divergence of the flow field? Any suggestions?
Yes. Sure.
What is the divergence of flow? That's the continuity, right?
Yes.
And what's the value of that?
That's it.
So what is this one? What is this?
You really want to do it, you just go. So opposite.
What is this?
But
So we've derived this before. That's continuity. Right? So it's 0.
but this
Yes? So then gradient dot UP, you distribute the derivative, this guy is 0, then you're left with what? U dot grad P, right? Okay, U and grad has nothing to do with its orientation integral. So you just integrate QQ minus this I times P. And what do you get from that? Kinematic. Okay, good. So we have this one. U dot grad Q. Yes, good. Okay. Other questions?
Cool.
So what we did here is a sort of bulk rheological measurement or macroscopic rheology, where we have a bulk system. We apply a shear flow and then we measure the force we're getting. So in the context of active systems or microscale systems, what's more relevant or interesting is microrheology. So this is again a way to measure flow and deformation of materials at the microscopic level. So here's a cartoon of how that works. So the blue particles are your suspension particles, whatever material you have, that's the microstructure. And you can just embed a passive bead. The size of the bead is typically comparable to the size of the microstructure. And you can apply force on this bead, then try to measure its motion, or you can specify a motion and say how much force I need to drive that motion. So in this context, we call this a probe particle, okay, or a tracer that's moving in a system. And these suspension particles are called a bath particle. So a typical experiment is done like this. So you have a sample here, and you have a particle that's driven by an external, in this case by a laser field, and that drives this particle forward, and then you can keep track of its position over time to quantify the response of this particle as it collides and moves through the bath particles. Okay, so why is that an interesting technique? For example, if you want to think about the rheology or the mechanics within a living cell, that's very small. You can't do bulk rheology, because bulk rheology, you're doing macroscopic measurement. You can't get at the scale of a single cell. So here's an example of the transport processes that are happening within a cell. Okay, video, let's play the video again. Before I do that, let's explain what's happening here. So this is a cell with untreated, and in the middle is treated by this chemical. So this is a myosin motor inhibitor. What it means is if you put that in here, you're gonna kill the activity of the molecular motors. And another way to kill this activity is to remove the fuel. So in the third video, there's ATP are depleted, so they again cannot generate active forces there. So if you look at the video, the motion that you're getting, or the fluctuations in the system, you see clearly that on the left, this one, wait. So you have the largest displacements in the first video, okay, where the molecular motors are active. In the second, you've sort of killed off some of these motor proteins. You have less displacement in these tracers. Similarly in the last one, because you have removed the fuel for these cells. So if you want to characterize these transport phenomena at this scale, what you have to do is you have to put a tracer into the active cell rather than doing a bulk rheological measurement. So here's an example of a macrorheological experiment. Again, this is looking at the cytoplasm of a living cell. And so here what you see, there's a little rod, which is magnetic. That's very small, a needle-shaped object that's being put into the living cell. Because it's magnetic, what you can do is apply an external field to rotate this particle, okay? And again, you can keep, in this case, you can keep track of its angular displacement over time. So that's what's being shown here on the right, the angular theta as a function of time. So you get different kind of curves depending on how you're doing the experiment. And from that, you can infer the rheological response you're getting. For example, characterize the viscoelastic behavior at the local microenvironment. Okay, so these kind of measurements have many applications, for example, in mechanical phenotyping, such as in cancer diagnostics. What you do is, what we know is, so if you measure the mechanical properties of a cancer cell and the normal cells, they're often different in terms of their elasticity and deformability. So that's a way of characterizing whether a cell, is in a cancerous state or not. So that's one thing one can do. People are trying to do that, which is a current topic that people are exploring about. So in microbiology, there's two sort of modes of operation. One is I just put this particle in the system without applying any external force on them. Okay, I just put it's called a passive microbiology, or just a tracer diffusion problem. So I put the tracer in there and then I just watch it diffuse through the matrix. Okay, so here's a sketch of how that works. So you have a tracer particle, it's going to undergo some displacement. It has to push around these particles.
Yeah, we have to find a good synthesis system of data.
No, I'm just talking about our operation here. There are other particles in the system. That's the material. So the material is composed of microstructures, which are particles or other small entities. No, we haven't talked about Acumatter yet. This is just, in general, a technique of measuring things. There's no Acumatter here yet. It's just a material. You put a bead in there and you keep track of its displacement over time. And the reason that you can use this to measure rheology is the following. So if you're putting a bead in a purely viscous environment, it's just undergoing diffusive motion, right? Then the mean square displacement is linear in time. What if you put a bead in a purely elastic medium? What happens? So the bead is in elastic environment. It takes a step. What happens? There's a spring force that brings it back. So on average, you can't go anywhere. So the mean square displacement is going to be a finite number, a constant. So if you were to plot the mean square displacement as a function of time, if it's diffusive, you have this red kind of shape. If it's somewhere in between, if it's viscoelastic, it's going to look something like that. So you can tell these materials apart by looking at a passive microbiological experiment. Okay, so so now let's come back to active systems and think about a tracer diffusion in active systems. So again, let's compare these three cases. One is a bead in just pure water. Then it's diffusive. Then the diffusivity you get a long times is just dt, right? That's the first term here. What if you have a suspension of other particles, but those guys are passive? So then as a tracer, if you want to go somewhere, you have to push around these particles, which means you get steric hindrance from these guys. So if you're actually in the dilute limit, if the size of the tracer and the particle are identical, you can calculate that. And the effective diffusivity you get in a passive bath is dt times this bracket, 1 minus 2 phi. Phi is the volume fraction of the bath particles. Okay, so one thing you notice is the diffusivity in a passive bath is lower because you're hindered by the presence of other guys. And this is a dilute result. When phi is small, you can calculate this exactly. Okay, so the question is, what happens for an active system? Now, these particles are all active except the tracer particle. In principle, you still have this steric hindrance, so we can write this out, and then we can define the difference as an active contribution, so on top of this hindrance. And as one may imagine, this gray particle is embedded in an active suspension. the red particles are active. So they're swimming around. They're going to push these gray particles around. So effectively, it looks like this gray particle is also active. So it's gonna convert some of this activity into the gray particle, which means if you were to characterize the diffusivity, for example, this part, it's gonna grow as the red particles become more active. So that's the packet number. That's more swim speed. Then that's gonna be transmitted to the tracer particle. And if the particles are active enough, you're gonna overcome this hindrance. So the total diffusivity is actually much higher than the diffusivity in just water because of the fact that you get a lot of collisions from these active guys. Okay, so you can solve these equations, again, making use of our model, only the dilute limit. If it's not dilute, you can just make use of Brownian dynamics simulations to understand how these things behave. All right, so that's a passive microbiology where the particle that you've embedded in the medium is just free. You're not applying an external force on them. Okay, the second mode of operating microbiological measurement is forced microbiology. It's still, you embed a particle in a suspension, but now you're applying external force to actively drive the particle into some direction. So historically, we call this active microbiology. But now, because we're working with active matter, so there's kind of a confusion of what do you mean by active. So in this context, we're going to call this forced microbiology, okay? Just to distinguish the fact that we're driving this probe here in a suspension of other particles. So in this case, you can define a viscosity just like you do in the rheometer. So the way to do that is, suppose you're saying that I'm going to operate this rheometer at a constant velocity of motion from the tracer particle. Then of course you have to apply an external force for that to happen. So you can get the net force on the particle that you're driving. You can use an effective viscosity in the Stokes drag law, okay, where U1 is the velocity of the tracer particle. And you can define an effective viscosity, eta effective, and you can define an increment in viscosity by comparing the force you need to apply compared to the force that you have in a just pure solvent. That's zeta, that's the coefficient, drag coefficient times the velocity. You can normalize that by the force you get in a solvent. So that's a dimensionless increment of viscosity, delta eta over eta, okay? In terms of this effective viscosity, that's the difference normalized by the viscosity of water. Okay, so that's a measure of viscosity at the micro scale, and we call that a micro viscosity. So if you think about active particles, active colloidal particles, here's a schematic of how that works. So you have a particle that's moving at a velocity u1, and what we want is to measure or to quantify the force that we need to apply in order for that to happen. So in the dilute limit, we can represent the suspension with the representative particle. That's only true in the dilute limit. And in that case, you only need to consider the pair problem, which is the interaction between the tracer particle that you put in there and a representative suspension particle, which may be active. So we can make use of the equations that we derived previously to study this problem. So let me try to share my screen from here. Okay, that's not working. Okay, I'm just gonna switch to the iPad.
Okay.
Okay, so let's consider the dilute problem.
Actions.
So in the dilute limit, again, we have a probe particle, okay? And we can represent the suspension with only a representative particle in the system, so we can only look at the pair interaction that's happening in the system. So if we define our coordinates, so we have the position of the tracer, x1, okay? And we have position of the bath particle, x2. If the particle is now also active, so we need to associate the orientation or the swimming direction to it, say q. Okay, so that's our configuration of the system. Then we can write down our Smolukowski equation. So now we have three coordinates we have to take care of. So again, in general, we write down our conservation law in this way. So that's a gradient with respect to x1. Then there's a flux for the tracer particle, okay? And a gradient for particle 2 in position space. But also particle is rotating because there's swimming motion. So there would be a rotational part for particle 2. Okay, that's our conservation law. And now we want to write down the fluxes for these particles. So particle 1 is the probe particle, right? So it is driven by a force, or in this case, we're specifying that the particle is moving at a fixed velocity. So that velocity would be u1 times the distribution. And because the probe particle, the size of the probe particle is comparable to the size of the active Brownian particles, so that means that particle also gets Brownian diffusion, right? So we can write, sorry? This is a pair distribution. That's right, it's two particles, it's a joint distribution, where P is, depends on position of 1, okay? Position of 2, and also the orientation of 2, and time, right? That's a pair distribution, but also including the orientation of the second guy.
Sure. Orientation at 1 with respect to 2 are the orientations of both the particles.
Orientation of the second, the first line is a Brownian sphere, essentially. We don't care about its orientation. So the second line is the active Brownian sphere, where we do care about its orientation now. Yeah. So we're trying to write this equation all the time because once you've done enough times, you can easily write this down, regardless of what kind of problem you're looking at. We read this stuff previously for one particle, okay, and also in the case with flow, now it's two particles, where one of them is Brownian, driven by the force. The other one is active Brownian. All right? So that's the viscosity for the probe.
This one doesn't go to singing. Can also have a one Brownian particle. It just considering V1 and 2.
Can we have
an old Brownian particle? Like say
you could, but then which means your particle is pretty large in reality, and much larger than the active Brownian does. Is essentially how much Brownian much you get is dictated by the size of your particle.
So what you have considering the particle to state like this?
Yeah, we're doing spherical particles. That's right.
Right. But the rotation of the particle
of this guy doesn't. This guy doesn't matter because this guy is active.
It doesn't work.
That's why we didn't include the rotational flux for one.
It is like something else.
If it's a ellipse, for example, then you have another term here that's the rotational flux of the first guy as well. But you see the structure of the equations, you can add that as you see what you need. So that's the nice thing about these equations. Great. Other comments?
It's in things is like something the probability density.
So now in this case, the probability density would be the probability density function. So whenever you're talking about the probability density function, you're talking about how do you fully specify the configuration of the system. Okay, how do you do that here? First, you have to tell me where the probe is. That's x1. You have to tell me where the active particle is. That's x2. But then you also have to tell me in which direction this active particle is swimming at. Okay, so which means the probability density function is finding the pair.
That's good.
With the probe at location 1, the active particle at location 2, and swimming in this direction q at time t. That's the joint distribution. Describes the full configuration, right?
Well,
Right. Other,
which means just that position.
yeah. Yeah, whatever is necessary to fully specify the configuration, they'll all appear in the conservation equation. Yeah, other comments? Okay. So we've written down the flux vector for the probe particle. And what about for the bath particle, the active particle?
Which one?
We know how to write that down. We've done that quite a number of times. So it has a swim velocity, right? In direction of Q. It has Brownian motion, so I call that d2t. Gradient of with respect to the position of the second particle, so that's graph 2. All right. But also it has rotational flux. And again, in this case, because an active Brownian particle is just rotating due to Brownian motion. So let's call that d2r. Okay. So now we've written down our conservation law for this pair in the system.
And ones that will apply.
All right. Any questions about this?
Useless.
The ATPs can also be under some kind of external force, right?
You could, if you want, you could. Then you add that here. Okay. But the context here is that we are trying to measure the behavior of the active suspension left alone by putting a bead in there, right? If you want to force the active particles for some other purposes, then you add the force there. Or you have some external field, then you add that too. Yeah. Make sense? Other comments? Okay, so let's jump back to the presentation. So the equations, I've written down the equations previously, and here the equation is slightly different because I've done a change of variable here by defining the position of the bath particle relative to that of the probe, rather than using the position in the lab frame, because that's really what's important. When you're driving the probe, you really care about where the bath particle is relative to myself, okay? So if you do that, you have a gradient with respect to the relative coordinate, and within that, it's the relative flux. Okay, that's easy to understand because you have to respect Galilean invariance. Now you're essentially if you're in the frame of the particle, the flux you get by looking at that guy is that the flux of this guy relative to myself. So it's a difference between the two, and the rotational coordinates is not changed. Okay, great. So far away. So if this bath particle is infinitely far away from the probe, what happens? It's a homogeneous suspension, okay? Because we're looking at the dilute limit, so which means the angular distribution is 1 over the angles in 2D or 3D, and the density is a system density at infinity. That's a number. It's homogeneous density. That's the far field. In the near field, at most, these guys, we're talking about spheres, so these guys can come into contact, okay? So that's the geometry of contact. The radius measured from the probe. In the contact radius, that's just the sum of the two radiuses, okay, radii. And at contact, particles cannot penetrate into each other, which means there's no flux going into this surface, right? So N here is the normal vector of the contact surface, and then the net flux is 0. So now we have fully specified our system, both the boundary conditions and the equation. Then we can try to analyze them. So we're not going to talk about how we solve these equations, but here's the result. So if I am plotting here the microviscosity that we defined earlier as a function of essentially the driving force on the probe, so how fast you're moving the probe. So what I see is a thinning behavior. So as you move faster, the probe, the microviscosity goes down. This is for the case of a passive system. So I've again turned off the activity of the blue particle. So what happens if you have active particles? So here's an activity Peckley is equal to 1, so it's moderately active. Again, here we're comparing the theoretical result, which is the PDEs we talked about, to a Brownian dynamics simulation. So for a moderately active system, we see we still have this kind of a behavior. Again, this is a repeating theme of looking at the regime where the external influence or forces or flow field is much stronger than activity. What happens always is you have to go back to the passive result. That's always the case. So in this case, that means when the external probe velocity is quite fast, I again can't tell whether my surrounding particles are swimming or not. So I go back to the passive. Only when the external driving force is small, I can see a difference between passive and active. So in this case, as I'm swimming, actually, my viscosity again decreases. And if I further increase my swim speed, my viscosity is lowered further in this regime. Again, in the high Peckley regime, I can't have a difference. What is increasing? In this region?
Yes, it has like
There's a slight another behavior here. Because in this regime, essentially what happened here, if you look at the Peckley is S, that's the swim speed, and the probe speed here, they're comparable in this regime. Okay, so you enter a funny sort of slight increase here before it then comes back down. So that's because of there's a multiple time scales that's happening in the system. And in this regime, yeah, you have a slight increase, but that's very, small, minor. So in general, what you see is, again, in this regime, we can't tell the difference. But on the left-hand side, you have I call this a swim thinning behavior. As you swim, it's easier to flow is a thinning behavior. It's sort of like a microscopic analog of the shear thinning behavior that we talked about before. So that's for active particles. And if we think about essentially why, in this context, why does the viscosity of active particles becomes less? So actually what you have to think about is to look at the number density distribution of the bath particles around the probe particle. And when the packet number is very small in this regime, you can do it sort of a Taylor expansion or a perturbation expansion of the density field. So you have an N0, that's the leading field that's obtained when the Peckley tends to 0. Okay? So when the packet number of the probe tends to 0, what happens? The probe is stationary, is fixed. And then you have a spherically symmetric density distribution of the bath particles. That's true regardless of whether they're active or not. Of course, when they're active, the density near the fixed probe is higher than far away. If it's passive, it's uniform. So that part of the microstructure doesn't contribute to the viscosity because it's spherically symmetric. So that's a tricky thing about this. And then you actually have to look at the non-symmetric part, which comes here in the order of packet number. And in this case, the reason that the swim thinning happens is if a particle is passive, if a particle is active, it's swimming. It can only be staying near the probe if it's pointing into the probe. If the active particle is pointing outside the probe, it's going to escape away, which means that you only have half of the orientational degrees of freedom of the bath particle that can be near the probe. Whereas for a passive Brownian particle, the orientation doesn't matter, which means you get the full orientation space, whether the orientation is pointing out or into the probe, they're all there. So you get more asymmetry for a passive system. So that's why the viscosity is higher there for passive compared to the active, okay? So that's a very intricate mechanism that happens in the context of microbiology. Questions? So far we looked at hard spheres, which means that we haven't considered the disturbance flow generated by either the active particle, right? As they move around in the fluid, they should generate disturbance flow, which we haven't talked about yet. So we neglected that part. And we'll talk about what happens when you have that in the next lecture. So I think we'll stop here and then come back in the afternoon.